Okay, so here is the last topic that we need to talk about when we're looking at blood histology and typing. And so this is basically related to the typing part. So we're going to go over blood groups and specifically focus on the ABO blood typing system. So basically what we have here are a classification system that is based on antigens that are present in your red blood cells or on your red blood cells, and antibodies that form in the plasma. So if we look at antigens, so basically these are going to be antigens that are located on the surface of the red blood cell. So I always kind of draw this red blood cell, and these are molecular markers. And in terms of ABO and RH, these are basically going to be glycoprotein or glycolipid markers that stick out from the surface of the red blood cell, and the blood group that you are part of is based on which antigen you have present on your red blood cell. Okay, so the blood group that you're in is based on the presence or absence of either type A agglutinogen or antigen, or type B agglutinogen or antigen. Okay, so it basically depends on which one is there or which one is not there. So if you have type A antigen on the surface of your red blood cell, well, you know what? Let's draw this a little differently. They are sticking out. What I'm going to do is I actually want them to be on the red blood cell. So here's my red blood cell, okay? And if I have A antigen present on the red blood cell, okay? So A antigen present, then I have type A blood, okay? It's as simple as that. It is important to remember that the antigens are located on the surface of the red blood cell, okay? Because when we get to antibodies, it's a little different. Now, what we have is antibodies that are located in the plasma, so they're going to be circulating around that red blood cell. And in the ABO blood grouping, these antibodies are preformed, meaning that you don't have to be exposed to the foreign antigen in order to form them. You are basically born with them. They tend to increase as you get older. So when you're younger, you don't tend to have as many antibodies. I believe the peak time for formation of antibodies is around 10 years old. Okay, so up until that point, you don't really have a full amount of antibodies in your plasma, but it peaks at about 10 years old, and then you maintain that level of antibodies. But the fact that they're preformed means that you don't have to be exposed to foreign blood in order to have them. They develop independent of that. Now, what's going to happen is that antibodies in the plasma are going to react against red blood cells carrying antigens that are foreign to that person. So basically, if your blood has A antigens, then if we took A antigen from someone else, type A blood from someone else, and put it into your blood, your body is not going to see that as foreign because you have the same antigen. Where we have trouble is when we have antigens that are not present on our own blood. We're going to see those antigens as foreign, okay? And we'll talk through some examples. It is important to remember that the antibodies are circulating in the plasma, okay? So antibodies are in plasma, antigens are on the surface of the red blood cell, okay? So let's walk through this. So this is what we have here. If we have type A blood, again, the type of blood that you have is named for the antigen present. Okay, so that's what all of these are going to do. So if we have type A blood, it means that A antigen is present on the red blood cell, it means the antibodies that are present in the plasma are going to be antibodies against the antigen that we don't have. If you recall, our only choices here are A and B. So we can have A antigen, we can have B antigen. We can have both antigens, A and B, or we can have no antigens, and that would be type O. I always tell students that's like a 0 because it basically means no antigens present. Okay? So if we had type A blood, it means that we have A antigens present on our red blood cell. It means that we're going to have antibodies circulating in our plasma against the antigen that we don't have. So we're going to have anti-B antibodies. Now with type B blood, again, the type is named for the antigen that's present on the red blood cell. So we have antigen B present on the red blood cell. And then we're going to have circulating in the plasma antibodies against the antigen that we don't have, because that's going to be the foreign antigen. So in this case, they're going to have anti-A antibodies. Now, in the case of type AB blood, so here's this scenario here, right? Now, what we have here is both type A antigen and type B antigen on the surface of our red blood cell. So since this blood type has both antigens present, it is not going to see any antigen as foreign. It has both of them. So it is not going to have any antibodies. It has neither anti-A nor anti-B antibodies. So no antibodies present in this blood type. Okay? Now, type O blood is exactly opposite. It has no antigens present on its red blood cells. As a result, it's going to see both antigens as foreign. So it's going to have both types of antibodies. It's going to have anti-A and anti-B antibodies. Okay. Now, on my YouTube channel, I've actually talked through a couple examples of these where we go through donor examples. And so I would highly recommend that you take a look at that. So basically what happens, where we run into trouble with this, is when we have blood donation. So basically what's going to happen is when you donate blood, so in the scenario of donating blood, okay, we're going to have the recipient's antibodies are basically going to react to the donor's antigens. Okay, so that's what's important. The recipient is the person receiving the blood. Their antibodies have the potential of reacting to the donor's antigens, the antigens that are present on the red blood cell of the donated blood. Okay, and those are the only two things that we need to worry about. So basically what we can't have happen in this case is we can't have A antigen pairing up with anti-A antibody. Okay? This creates what's called an antigen-antibody complex. Okay? And this is not good. Okay? So we don't like this. This creates this scenario down here. So basically what happens is the blood vessels, the red blood cells and the antibodies are going to clump together and then eventually break up the red blood cells and destroy them. And that process is called hemolysis. But this complex that forms creates kind of a clumping type of reaction in the blood, and we call that agglutination. Okay, so agglutination happens, and this is bad. This is not something that we want to have happen. Okay, so this is bad. So what we don't want to have is the recipient's antibodies. Let's say we have type B. Well, I was going to walk you through a couple examples. Okay. let's say that we had, in the recipient's blood, we had type B blood. Okay, so here's our recipient. Okay, what's important here? The recipient's antibodies. So the recipient has anti-A antibodies. Okay, now let's say this blood type here is our donor. Type A blood is our donor. Could we give type A blood to type B blood? Well, let's look and see. The donor's antigens are what's important here. So this donor, type A blood, is going to have antigen A on its red blood cells. Can we have this? Can we have anti-A antibodies and antigen A present together? Well, we can't. It's going to form this. It's going to form this antigen-antibody complex, and it's going to result in agglutination. So no, this would definitely not work. Okay. So that's an example of a blood transfusion that is definitely not going to work. Now, let's talk through another one. I'm sorry that my slide is getting so crazy here. Let's try to clean this up. So what would happen if we had, let's see. Okay, now let's say that we have type AB blood as our recipient. Okay, what's important on our recipient is the antibodies. So here with type AB blood, these guys have no antibodies in their plasma. Let's say we keep type A blood as our donor, and we know what's important here is antigen A on the red blood cells. Now, are we able to give type A blood to type AB blood? We are. That would work. So we could say yes, that is a transfusion that will be fine. In this case, it's going to work because the recipient has no antibodies present to react to the donor's antigens. Okay, so the reason why this transfusion would work is because there are no antibodies in type AB blood in order to agglutinate the A antigen on our donor blood. Okay, let's work through one more, and I'm just bear with me. I'm gonna try to clean this up. And I want to give you one more scenario. Okay, so let's say that we have a little different scenario, and let's say this type O blood is our donor. Okay, and let's make type B blood our recipient. So here's our recipient. Okay, so what's important in our recipient blood? The recipient's antibodies. So let's highlight that. Type B blood is going to have anti-A antibodies. Okay, what's important in our donor's blood? The antigen. So here's our donor blood. What kind of antigens do we have here? We have type O blood, which means that these guys have no antigens present on their red blood cells. Okay? Are we able to do this? Can we give type O blood to type B blood? Yes, we can. That's going to work because the anti-A antibodies don't have any antigens on the donor's red blood cells to agglutinate. So there's going to be no agglutination. So yes, that is going to work. Okay? So I would highly recommend that you watch my other videos. I work through a few more problems of blood donation. It is important to know who can donate to who. And why, actually. That's a great test question. Okay, and so just a little bit about the genetics of ABO blood grouping. So A and B are going to be codominant alleles, and type O is going to be the recessive. Okay, so we're going to see A and B, and then type O is going to be our recessive. But what we have here is like if we had an A allele from the mother, an A allele from the father, the genotype is going to be AA. The blood type of the offspring, or the phenotype, the physical manifestation of the genotype, is going to be type A. In this case, we've got A and B, and since they're codominant, they're both going to be expressed. In this case, we've got an A allele from the mother donated, a recessive O allele from the father, and that means since that O is recessive, we're going to have type A blood as a result. And then if we come down here, we've got a recessive gene, or a recessive allele from the mother, recessive allele from the father, and then that's how we would end up with type O blood of the offspring too. So there's a couple examples of Punnett squares to go through to see how that would play out with alleles from the mother and father too. There's one more blood grouping factor that we need to talk about, and this is Rh factor. And so basically this is another antigen. Like I said, there were only A and B antigens, but there's actually one more that we're going to talk about, and that's going to be Rh. This is another antigen that is present on red blood cells. And what we see with this is about 85% of Americans are Rh positive. And if you are Rh positive, it means that you have the Rh factor or antigen on your red blood cell. Okay? So positive means that it's there. If you have Rh negative blood, that means that your red blood cell does not have the Rh factor. And if you are given blood that has the Rh factor, your blood is going to see this blood as foreign, and it's going to develop antibodies against this foreign antigen. So now this blood is going to develop anti-Rh antibodies, okay, that are going to then attack any Rh antigen that they come into contact with. Okay, the thing that's different with this is that it happens after exposure, okay, as opposed to preformed like the ABO. So it is not preformed. It only happens after you've been exposed to Rh positive blood. So after exposure to Rh positive blood. Okay, so where we, oh, this is what it looks like too. So here's our red blood cells. If you're Rh positive, you have that Rh antigen on your red blood cells. If it's negative, then you don't have the Rh antigen on your red blood cells. Where we run into trouble with this is with pregnancy. And so in this case, if we have an Rh negative woman and an Rh positive man conceiving a child together, it is possible that the fetus is Rh positive. Okay, so the fetus is going to have the Rh antigen on their red blood cells. The mother does not. So as a result, the mother's blood is going to see the fetal blood as foreign. The mother's blood is going to form antibodies against the Rh antigen on the fetal cells. So against Rh antigen on fetal red blood cells. Okay? And it's going to attack. So we have the process of agglutination, which is basically, what we see here. So this is going to happen. So we're going to end up with a situation where we have hemolysis of the fetal red blood cells, and it could actually destroy them. So there's a test that they do for this. And if it's determined that you, as a mother, have Rh negative blood, they're going to give you an injection. It's called RhoGAM. And that's an injection that basically is going to suppress your body's ability to form those antibodies against the fetal red blood cells. So it basically takes care of this, and we don't really have trouble with this anymore. But this is a scenario where you have issues with Rh factor with red blood cells. And typically the first pregnancy, they still treat it. It's not that they don't treat it, but my understanding is the first pregnancy is not quite as serious as the second pregnancy, because the mother's body doesn't have quite enough time to generate the number of antibodies. But I think it's still a problem, and they will still give you a RhoGAM injection too. So, okay. So just when we talk about donors and recipients, we have what's called the universal donor, and this basically means that they can donate to anyone, and this is specifically going to be type O blood, because, in fact, it's going to be type O negative, because it has no antigens present, right? It doesn't have A or B or Rh antigen. So it has no antigens for the antibodies of the recipient to react to. So therefore, type O negative is considered the universal donor and can donate to anyone. Now the universal recipient is going to be type AB positive. So look at these guys. Your blood type is named for the type of antigens you have on your red blood cell. So these guys have all of them, right? They have all the available choices. They have A antigen, they have B antigen, and that positive tells us they have Rh antigen. So since they have all antigens on their red blood cell, they are going to have no antibodies. They don't find any antibodies, or I'm sorry, they don't find any antigens foreign because they have all antigens. Okay, they have all the choices. So they have no antibodies. So as a recipient, we know that we have donor antigens, right, which are going to recipients' antibodies. Okay, so what's important here is the recipient's antibodies. So in this case, we have no antibodies to react to the donor's antigens. Therefore, type AB positive blood is considered to be the universal recipient. They can take blood from anyone, which is a great place to be. So in terms of Rh factor, what we see with Rh factor is that negative blood can donate to, if you're Rh negative blood, you can donate to Rh positive blood, and it's going to be okay. Or you can donate to Rh negative blood, and it's going to be okay. Okay, so negative blood can donate to either. If you are Rh positive, you can only donate to Rh positive blood. Okay? If you try to donate to Rh negative blood, what's going to happen? These guys don't have the antigen. Okay? They don't have the Rh antigen. That's what that negative means. These guys do have the antigen. If you try to give this blood, the positive blood, to the negative blood, it's going to see, the negative blood is going to see the positive blood as foreign, and it's going to generate antibodies against that foreign antigen. Okay, so this does not work. We'll say no, that does not work.